all. This is Dr. Mobeen Sayed from drbeen.com. Welcome to one more show. So today's discussion is really interesting. This is the case and the technology of a teenager girl in United Kingdom who had an incurable acute lymphoblastic leukemia of T cells, and she is now cured. And fingers crossed, praying for her that she stays cured. It is 6 months into the management and she's cured when she had reached a point of the doctors saying, we have no other option left. So let's look at the technology behind this. And congratulations to Alyssa, who is the girl who is cured. Dr. Professor Wasim Qazi, who was her managing team's lead, and Dr. David Liu, who is the inventor, and his team is the inventor of the base editing technology that was used for the first time in the world on a human being and helped cure them. So I really believe that children who are being born now, some of them are going to live 150 or 200 years. I hope the world resources are good to nurture them and to offer them a good living, but health sciences are progressing fast. So let's look at it. So this is drbeen.com. In the description of this video, there is a link to this page that you're seeing. And here, for a very limited time, you have access to 900 lectures, medical lectures on Dr. Bean for only $67, one-time payment. It's not a recurring payment. Then here is the news, and I'm going to discuss this in detail, the technology. This is the news. Base editing, revolutionary therapy clears girl's incurable cancer. And so the girl's name, Elisa. Then there is another news. Living drug offers hope to terminal blood cancer patient. This was for CAR T technology. Today we're going to talk about CAR T and we're going to talk about base editing. So this was CAR T technology, again, a wonderful technology. Base editing kind of develops on top of that or is added to CAR T. Then here is the link to understand what CAR T is. I'm going to provide an overview as well. And then there are some more studies. This is important: programmable editing of a target base in genomic DNA without double-stranded DNA cleavage. So this is that Dr. David Liu and his team, their work, and this was just done 6 years ago. And today, or this month, today meaning this era, this time frame, a girl has been cured using this technology. So who is Dr. David Liu? If I can very quickly show you, here is Dr. David R. Liu. He is professor at Harvard Broad Institute and HHMI. Our lab integrates chemistry and evolution to illuminate biology and enable therapeutics. So this is their paper. Then this is the hospital where this treatment was done. So Great Ormond Street Hospital, or GOSH Hospital. Although the CAR T technology and the base editing was done here in the US, I believe at this time this technology is available in the US. So the cells are actually sent here. Then these are modified or edited here and then sent back. Then some more references. What is CD7? That we'll discuss. CD19. Then pathogenesis and prognostication in acute lymphoblastic leukemia. That is what she had. And so on. There are some more. And there is a video
with
here
a
as
TCR.
well. That is a wonderful video to explain the mechanism of CAR T if you wanted to dive deeper in that. Then some more diagrams here. and about the cytotoxic T cells that we will be talking about today. So with this all, let's start our discussion with my diagrams. So these are gifts for humanity. They're continuing. And my apologies, as you cannot see my face, this is a different recording software. So here is a plan for the talk today. First of all, we'll look at the case of this teenage girl with incurable leukemia who has been now cured under monitoring though. Then researchers and physicians who I just shared as well, who they are and how did they help. Then what are the CAR T cells, then pros and cons of CAR T intervention and base editing as well, especially in context of this little girl's case. So here, base editing, what was done? So Alyssa is a 13 years old teenager. She was told last year or diagnosed last year that she had leukemia, T-cell acute lymphoblastic leukemia. What is that? We have been talking about T cells and B cells and acquired arm of the immune system. So this is the cancer of the T lymphoblasts, where these lymphocytes, instead of becoming mature cells to help the patient fight infections, instead of that, they just keep making more cells, and that's all they do, just make more cells. So the result is that these cells fill up the bone marrow, then they spill out of the bone marrow in the blood, they go to liver, they go to kidney, they go to spleen, they go to other parts of the bones, and they are not really capable of fighting very well because these are still immature cells, but they're just filling the body. So at some point, patient is not able to handle infections. That is one cause of death that can happen. And then, of course, the cells filling up various body tissues and damaging them is another. There are other reasons too. Now, for her, all treatments had failed. She was given chemo that failed. She was given a bone marrow transplant that failed. And she had no other option other than doctors saying that let's make you as comfortable as we can. All other options had been exhausted. Then they decided that, hey, how about we try the living drug or CAR T cells? Again, they leave the CAR T at this time in your mind. I'm going to explain it. And then they also decided that how about we use base editing as well? This is the first time base editing is used. So 6 months later, 6 months after she was given this intervention, her cancer is undetectable. She would have died if this therapy was not used. She is the first patient ever to use base editing on CAR Ts. The CAR T cells destroyed Alyssa's cancer cells and healthy T cells. So imagine that we made cells, or the researchers, they made cells that were injected from outside, the soldiers that came in from outside. These specially trained designer cells were specific for Alyssa's cells. And they went in, and these soldier cells, they went in and they wiped out all of her cancerous cells, T cells, and healthy T cells. They just indiscriminately killed them all. And once these were all killed, then a bone marrow transplant restored new T cells for her, and those new T cells are now cancer-free. They are new. Now, when her T cells were destroyed by these external cells, the CAR T base edited cell that were injected in her, when they destroyed her own T cells, then she was left vulnerable to infections. Of course, because there are no T cells to fight infections, or there are no T cells to fight a cell that is infected. Cytotoxic T cells' function is to destroy other cells that are infected. So she was vulnerable to infections. And so she stayed at hospital for 6 weeks so that she's not exposed to anyone with infections. So at 3 months after the management, there was a little scare because there were signs of cancer once again. But this is what we mean by the living drug. Those soldier cells were also present. So maybe the cancer tried to bounce back and these cells very quickly went and killed them again. Right? So they're just sitting there and saying, all right, if we see a cancer cell, we're going to attack it. So 6 months later, she actually is clear. So Professor Wasim Qasim, shown here, was part of the team that developed the base editing therapy. So this is Dr. David Liu that I showed you. Programmable editing of a target base in genomic DNA without double-stranded DNA cleavage. That is the technology that was used. So now let's very quickly understand what was happening. So this is my diagram of our immune system. It is not fully complete with beautiful cells because I'm still drawing those cells. But if you see here, we have these stem cells. Our whole body starts with 2 cells, correct? One from the mother and one from father. And then those 2 cells become these trillions of cells. And then every cell somehow knows who I am going to be. I'm going to be a muscle or a bone and exactly at the right location. So the original parent cells are called stem cells. Stem cells by definition are the cells that can make more of themselves and make cells that can become other cells. So here, imagine this is a stem cell. This is a multipotent hematopoietic stem cell. Multipotent means it can make many kinds of parent cells. So it's a parent cell that can make further parent cells. And these are all hematopoietic. That is, they are going to make blood cells. So here, then we have a common lymphoid progenitor. So it's a blast cell that is going to make lymphoid cells. So lymphoid cells being natural killer cell, T cell, and B cells. Then, of course, there is a common myeloid progenitor that makes all of these cells. I'm not going to go in that detail. Here, what I would like you to focus on is this is the cell line that was problematic in Alyssa's case. So her T cell lymphoblast, that is a parent that would give rise to T cells, that cell was cancerous and was just making its own copies. So we had to destroy, or the doctors had to destroy, Alyssa's T cells, especially cytotoxic T cells. Remember, T cells are of many types. There are helper T cells, regulatory T cells, then the cytotoxic T cells. So her case, cytotoxic T cells. And cytotoxic T cell in our discussion over here will be identified with CD7 on them. CD stands for cluster of designation. That means a cluster of proteins that are uniquely identifying a particular cell type. So CD7 and cytotoxic T cells. So here, a normal process of the cell interaction with a sick cell. That sick cell may be a cancerous cell, or it may be a cell that has a virus infecting it. It may be a cell that has a bacteria inside or fungus inside. A sick cell. So here in our case, this is a cancerous cell. And here is a cytotoxic T cell, CD8 positive, CD7 positive T cell. So if you see here, the T cell comes over, attaches with the sick cell with the MHC1, major histocompatibility complex 1. So you can imagine that there are 2 proteins, one on the sick cell and one on the fighting cell. And through these 2 proteins, these cells interact. The sick cell present an antigen on MHC1. So what happens is when that antigen is seen by the cytotoxic T cell, then the cytotoxic T cell says, you know what, I'm going to attack you. It seems like you're sick. I'm going to destroy you. However, our immune system is very smart. It doesn't just willy-nilly go and start destroying cells. If that happens, it would just destroy us in a day and be done. I'm exaggerating, but it will destroy us. These are what autoimmune diseases are. So here the T cell says, fine, I see something offending here, but I'm not going to become active before I double check. So there is co-stimulation needed. The co-stimulation here is, for example, done with these two further handshakes. So there is a CD28 protein on the cytotoxic T cell, and then there is a B71 or B72 or CD80, CD86. These are protein names. These are unique proteins. So these interact with each other. So you can say that this second handshake is sort of a verification, confirmation that yes, there is a problem and let's fight. Sometimes this second or co-stimulation, this second handshake can actually be helped or replaced by the cytokines released by nearby helper T cells. So again, immune system is making sure that it double checks before it kills a cell. Now, what if this cancer is of the T cell itself? So imagine, what do we need these cytotoxic T cells for? We need these cells to go and destroy the cells that are sick. What if this cell itself is sick? Now we have a problem. That is what happened in Alyssa's case, and that is leukemia. So what if this T cell itself, or B cell, or helper T cell, any of these cells, when they become cancerous, we call that leukemia. So in Alyssa's case, her cytotoxic T cell, CD8 positive, CD7 positive cell had become cancerous. So she had acute lymphoblastic leukemia of T cell type. So now they are out of luck because her own cells that are protective cells are cancerous. So who is there to protect? So now let's bring in the technologies that helped her. So of course they tried chemotherapy on her, but these cancerous cells were so aggressive they would bounce back. They tried a transplant on her, but still some cancerous cells were hiding somewhere and they bounced back. So then they said, you know what, as a last resort, let's try CAR-T formation. So the CAR-T technology is called the living drug, or artificial receptor drug, or chimeric antigen receptor T cells. Chimeric antigen receptor. Chimeric means when more than one things are fit together to create another functional protein complex. So here, normally what they do is, to make a CAR-T cell, they take a patient's blood. Then they take out the T cells from that blood. So they are taking, in Alyssa's case, because her own T cells were cancerous, they could not take them, so they had donated cells. But in case of in other normal cases, what happens is they take the patient's own T cells and modify them. So in Alyssa's case, there was a donor. So they take the blood and then they isolate and filter out the desired cell, in this case cytotoxic T cell. So here is a cytotoxic T cell standing saying, hey, where are the other cells gone? That is because we have isolated this cell. So once we have isolated this cell, then what we do is we want to make changes to its DNA. We want to make it to become a specific fighter cell. We want it to go and attack a specific kind of cells. So now to do that, these cytotoxic T cells, as I showed before, have receptors on them called T cell receptors. These are the T cell receptor through which the cytotoxic T cell will bind with the sick cells. However, we want these specific T cells that we brought out to go and attack a specific kind of a cell. So what we have to do is we have to edit their receptors. We have to change their hands so that they can fit a specific cell surface or surface protein. So how do we do that? We create the new DNA that would help produce those new cell receptors, and then we pack that DNA in a virus, harmless virus. And with COVID discussions, we have been talking about these technologies for a long time. So then this little virus is introduced in the T cell, and that virus would bring this DNA, and that DNA will be integrated into this T cell's genome, and now we've gotten an edited T cell. And now what have we edited in this? We can do all kinds of changes. However, in this technology, what genes are changed are the following. Number one, imagine here is an antibody. So there's a lot of work that goes in it. So you bring in the cell that you need to modify to make them into specific soldiers. You also bring out the cells that are the target. So you then figure out what is unique on this target cell so that we can hone into that target. You don't want to go and attack the whole body. So you have to figure out what is unique on the surface of these cancer cells. So you bring in the cancer cells out, you figure out a way to bind with them, and you create an antibody. Then you know that on the surface of the antibody, or on the binding region, variable region of the antibody, that is the portion that will bind. So you take that genetic material that makes the variable region of the antibody, that antibody will be able to bind with the cancer cell's specific protein. You take that gene. Then this is important that the receptors, if I go back here for a second, when this receptor engages, then inside the cell is wiring. Just like if somebody rings a bell and then there is wiring and then there is a bell and that bell rings and somebody responds. So inside the cell, there are proteins that are called second messenger system or signaling system. Those proteins are then attached with the second messenger proteins and so on. This is just like if somebody listens to a doorbell and then goes to the door, that person is a second messenger protein, but the doorbell and the wiring itself is a signaling system. So what they did was they artificially built a new receptor and they attach that to the signaling molecule. So that is what they are creating. This is a designer cell. They're making it in the lab. So now they have the signaling molecule attached to this variable region, which would act as a new receptor. This is like giving somebody a new hand, taking their hand out and giving them an artificial hand that can do some powerful moves. And then they also can add, depending upon what CAR T generation of technology, there are 4 generations, they can add even more molecules and proteins, gene in this structure, which would allow, for example, the cell to make more cytokines or to have more regulatory proteins on its surface. So they can design the cell as the need is. So the binding is a need, then signaling within the cell is a need, then cytokine production is a need, then more regulations or regulatory proteins are a need as well. So when that system runs, then this T cell that is now designed will make an artificial receptor on it, which will have this CAR T technology based protein. It will have a binding region that will bind with binding new artificially created binding receptor that will bind to a specific cell protein, and it has a signaling molecule inside that will become activated. It has cytokine molecules, and now we have a beautifully ready cell. It doesn't need double checking. It doesn't need co-stimulation. All molecules are attached just with this stimulation system, and it will, as soon as you bind here, the cell will become active. However, please remember, this technology is at this time expensive. So anywhere $420,000 or more or less, that is a kind of technology. It is expensive. Secondly, there are issues. For example, there have been deaths in the studies because of the neurological issues, especially cerebral edema. Then, because we add the molecules to produce cytokines, sometimes cytokine syndrome occurs. Actually, if I go back here for a second, this gentleman here, he was given CAR T technology. He also survived. He was also out of luck, but he had to stay in ICU for weeks after weeks because he almost died because of the neurological outcomes. So this technology has its risks as well, although with more and more generations of designer cells, these risks are slowly reducing. But still, we do not know how the neurological issues occur. Why does the inflammation and edema of the brain occurs? So, now in the case of Elisa, and in case of CAR T cell technology in general, there is still a limitation of using this. The limitation is this. You can introduce CAR T cell, for example, let's say for a T cell, or a B cell, or a red blood cell, let's say sickle cell. And you can kill them, and hopefully then you can do a bone marrow transplant, or you can just infuse more cells. So in the cases of blood cell issues, you can handle them with CAR T technology and wipe them out and recreate them. But you cannot do that to every tissue. You cannot send CAR T cell in and say, go ahead, there is a cancer in the lungs, destroy the whole lung, and we will just make a new lung. That's not going to happen. So CAR T technology is not universally available for every tissue. What does that mean? What that means is, imagine if somebody has lung cancer. Then they will have to spend even more time to figure out what is the very specific markers that are only on the cancer cells and are not on the rest of the lung healthy cells. And of course, that would take time. That will become more expensive as well. But we will reach there. At this time, CAR T technology is mostly used for leukemia-like situations where even if you eliminate healthy and cancerous cells, all of them, indiscriminately, you can still then come up and create those cells with transplants or with infusions. So now the next technology, base editing. This is Dr. Liu's work and his team's work. Base editing is another technology. So what happens is, when we are doing double-stranded DNA editing, that is CAR T technology, where you change the genes. When you're doing that, so normally what happens is there are lots of insertions and deletions that are randomly introduced at the points of editing. And I have just shown this break like this. Editing is usually done with one overhang on one side and the other overhang, and then you make the complementary sides. So there's a lot more complexity than what I'm showing here. But the basic idea is that when you are editing a DNA and removing a part of a gene and replacing it with another, or amending it and fixing it, it can become inefficient and error prone in some cases. So for example, if you want to change these bases are the nucleotide bases or nucleosides that are present in the DNA, then having all of this extra risk, and then the cell itself responds to this genetic change, that response itself can cause insertions and deletions, and that has inefficiency and problems. So what they did was, Dr. Liu's team, they created a new technology where they do not alter or edit the DNA out and replace it with anything else. What they do is they have created a new way to only change one base at a time without cleaving the DNA, without cleaving the backbone of the DNA, without replacing a part of the whole DNA. That is not needed, just a base editing. So this is the base editing technology, and what they do is they have created a new system with CRISPR plus Cas9 plus cytidine deaminase enzyme, which is reprogrammable. We'll do a deeper dive at some point. I want to stay within the focus of context of this little girl's cancer. So this base editing on top of the CAR T was done for the first time for this case. So what did they do? They took the cells, the cytotoxic T cell from a donor. They made them CAR T. That means they gave them this little receptor, artificial receptor, that would attack Elisa's cytotoxic T cell, and it would attack the CD7 part. CD7 is unique for a cytotoxic T cell. So what they did was, remember the soldier cells that are going in are also cytotoxic T cells? So how did they handle this? First, they modified this donor cell to recognize Elisa's cytotoxic T cell by recognizing CD7. Now the problem is, then these anti-cancer cells would recognize themselves as a CD7 as well and start killing each other. So then they removed the CD7 from these donor cells so that they don't start fighting with each other. I wish we learned that as well. Then T cell removed to prevent attack on the other side. Remember the T cell receptors that will bind with MHC1 and then attack the cell that is showing an antigen? Because these are donor foreign cytotoxic cells, for them, whichever body you put them in other than the actual origin, the donor, these T cells are going to attack. So they actually removed their original receptor from them so they do not attack anyone. And they left a designer receptor on them. So they remove their CD7 so they don't attack themselves. They don't fight with each other, but only attack the CD7 of Alyssa's cells, which is CD7 cytotoxic cells, and they remove their own MHC cells. Then they did further base editing to allow the DNA changes to protect these cells from chemotherapy drugs. So they kind of gave them a cloak on them. The result was these cells, then they made a lot of copies of these cells here and they introduced them in her body. So then the result was that these designer cells, when they went into Alyssa's body, they attacked her CD7, which was a marker on her T cells, cytotoxic T cells, which were cancerous. They did not attack each other because they did not have CD7 on them. That was removed, edited out. Plus, they did not attack any other cell through MHC1 binding because MHC1 receptor was removed as well, or the T cell receptor was removed too. And then they released cytokines. Remember, they released perforins and granzymes. Perforin punch holes in the target cell. Granzymes go into the target cell and trigger the destruction of it. So these cancer cells started dying. So we sent in cytotoxic T cells to kill the cytotoxic T cells, but we modified the going in cells that they do not recognize themselves as cytotoxic. Their CD7 was removed. That was the technology which finally resulted in her curing fully, praying that she stays cured, and then these kind of technologies will be used for so many other patients. We will have so much advancement in medicine that children who are being born now, they would have very good, long, healthy lives. And of course, we will reap the benefits as well. In a few years, these technologies will become more common. So thank you very much for listening in. In the description of this video, if you like this kind of a work, there are links in the description. You can buy me a coffee. You can become a patron, $5 a month, and become a patron. I would love that. Or you can use PayPal, or there are other links as well that you can use to support this work. Thank you very much, and I would see you again.